Hello, everyone. My name is Roland Faure. I'm a second-year PhD student in France and Belgium, and I'm very glad to be presenting to you today the work I've been doing during my PhD about separating strains in metagenomic long-read assemblies. So if you look up microbiomes on Google Scholar, you will find many great articles. And these are just a few that I found in the top scoring Google Scholar query. But all of these articles have a common shortcoming, in that they speak only about species level diversity, species level interaction, and they do not go to the level of the strains, the different strains that can be found in a microbiome. And today, large knowledge gaps remain concerning our understanding of microbiome, because microbiomes are most often analyzed only by looking at which species are in the microbiome and not which strains. And so the work of my PhD is really trying to get down to this strain level to improve our understanding of microbiomes in general. So in particular, my work focuses on genome assembly, on metagenome assembly. So when an assembly is computed by an assembler from reads, from erroneous reads, if the divergence between several regions is small compared to the error rate, the assembler will very often discard the divergence as error rate and assemble only one sequence. And that is why assembling strains is challenging, because most often similar strains are very highly similar at the level of DNA, and so assembler will only assemble one mosaic genome, which is a mix of all the strains of these species that are present in the metagenome. And this work is made particularly difficult by the fact that we generally don't know the number of strains that are present in the metagenome. We don't even know if there are several strains or just one. Moreover, we have the problem that all the strains are not covered equally, generally speaking, and you can have much more abundant strains and much rarer strains. And this is one of the main reasons that microbiomes are only studied at the species level. It's because we struggle to assemble the strains separately. So today I'm going to speak mostly about long-read assembly. And there is already a little state-of-the-art concerning assembling strains separately using long reads. And I would say the two most famous tools are Strainberry and Strainy. But I'm going to try to move a little bit beyond that. So the method I'm presenting to you today is named HairSplitter, and the goal is to go from an assembly generated by any means, by a normal assembler, and HairSplitter is a method that will look at each config of this assembly and split it into several versions if there are actually several versions of the config, one for each strain. And the name of the method, HairSplitter, was found by my supervisor, Jean-François. And a HairSplitter person is someone that makes very fine distinction in reasoning. And the tool here, the method of HairSplitter, in this case, makes very fine distinction in assembly. And that's why it's called like this. Okay, separating the strains is easier said than done. So let's try to do this together. And I'll show you how HairSplitter proceeds to separate the strains. So can you see several strains here? Well, the first intuition that pretty natural is to call variants. If there are several strains in this pileup of reads, there are probably variants distinguishing one strain from another. However, this is not sufficient. And as all of you that already did variant calling sometime in your life know variant calling is not an exact science, and there are often many false positives. And this is especially true in this case, because when you have a metagenome, you have this uneven coverage between the strains. You can have very rare strains. So a basic variant calling produces many false positives. And now is coming the key idea behind the method of HairSplitter. The key idea is to keep only robust variants. And robust variants are determined from the fact that there are several variants along the contig that separate the reads into the same groups. So for example, if you take the blue separation here, you have 3 different variants that separate on one side read 2, 5, and 6, and on the other side read 1, 3, 4, 7, 8, and 9. And this couldn't happen by chance. If these variants were false positive, there would be no reason that several variants along the genome separate the reads in exactly the same way. This is statistically unlikely. And so HairSplitter key idea is to find this robust variant by finding the recurrent pattern of SNPs. And here it found two partition of reads corresponding to 6 variants all in all. And HairSplitter will consider from now on only these variants and discard all the rest as false positives. So the next steps of the pipeline are to compute pairwise distance between reads. So the distance is computed based only on the robust variants. Once the pairwise distance is computed, a graph can be built using the k nearest neighbor to build the links of the graph. And as a third step, the graph is clustered. And here, the clustering algorithm, so is Chinese whispers algorithm, and it produces here 3 different groups corresponding to 3 different groups of reads, corresponding to 3 strains that were actually aligning on this contig. So once the 3 groups have been determined, each group of read is assembled separately and correspond to 1 strain. So that is the theory behind the method of HairSplitter. So it has been implemented in the software named HairSplitter. And does it work? So to evaluate that, we first began with simulated data. And more specifically, a mix of 2 to 10 E. coli genomes with simulated reads. There are many metrics that can be used to determine if an assembly is well strain separated or not. I will use only one here, show only one here for the sake of simplicity. It's the 31-mer completeness. 31-mer completeness is very basically the percentage of the k-mers of the solution, which we know in this case, that are found in the assembly. So ideally, 100% of the k-mer of the solution are present in the assembly if the assembly is perfect. Now we'll just choose this metric, but of course, there are other ways also to measure the quality of the assembly. So how did it work? So here are some results. On the left with the simulated nanopore sequencing, and on the right with simulated hi-fi sequencing. And in both cases, we see that HairSplitter improves very much the 31-mer completeness of the assemblies upon Metafly. And it does so much better than a strain barrier or straining, especially when the number of strains is high. With 10 strains, HairSplitter is really much better than strain barrier and straining at recovering all the different strains. So that was good, but then we wanted to test it on more real data. So we went up to mock data sets. So this is the Zymobiomics gut microbiome standard, in which we know there are 5 E. coli strains. And once again, HairSplitter really performed well compared to our Stringberry on all three kind of data that were available, but especially on the low quality nanopore. And so the last level of difficulty that we wanted to try was real data. So we took this data from a paper that was published 1 year ago, where 5 Vagus strains were sequenced using nanopore barcoded reads, which meant that the reads could be separated by strains and the strain could be assembled separately. So we had the solution. But then we chose to ignore the barcode to obtain a mixed assembly of the 5 strains, to obtain a mixed sequencing of the 5 strains, from which we built an assembly from which we separated the strains. And in this case, again, 31-mer completeness was much better in using HairSplitter than using the state-of-the-art Stringberry. Okay, so HairSplitter works very well. I'll just finish on a limitation that you have to keep in mind when you use HairSplitter, is that the separation of reads is done only locally. So that means that if you have at one point a region where the 2 strains cannot be distinguished, it will create such a graph where the resulting contigs are a bit shorter, because HairSplitter has no way of phasing the strains across collapsed regions. So it means that the N50 can decrease significantly if you use HairSplitter. That's something to keep in mind. So just to conclude, HairSplitter can reconstruct a strain-aware assembly from a draft assembly. You can use any type of read to do that, including high error rate reads. And it works on metagenomic and maybe also on multiple assembly, even though we didn't test it already. And the best part of it all is that it is available online on GitHub, and please feel free to download and use it and raise any issue you want. So thank you very much.